Morning, everyone. Feel free to move up towards the front if you like. There's lots of room, and we're a friendly bunch. I won't make you come up on the stage if you come up too close. I'd like to welcome you. I'm Michael Grant, Memorial University of Newfoundland. On behalf of myself and my co-chair, Amy Gilgrass, who's recently come from beautiful Hamilton and moved to beautiful Halifax. She's at Dalhousie University. I'd like to welcome you to this session on vaccines and other approaches to prevent STBBI. Right at the start, I'll say we're not going to discuss PrEP in this session because there's a separate session later on completely dedicated to PrEP. And we have 4 speakers. We're going to let all 4 speakers deliver their presentations and then have questions at the end of the session. So save your questions till the end, write them down in case you don't remember through the first three speakers. Neither Amy or I have any relevant conflicts to declare. I want to just put a little context to the talks before we get started. It's a little hard to believe it was over 40 years ago that HIV was discovered and identified as a cause of AIDS and characterized. Just rapidly, there was a blood test or an antibody test developed to screen the blood supply. And the U.S. Health Secretary at the time declared an AIDS vaccine will be ready for testing within 2 years. And there was a lot of optimism that we would have an effective vaccine. And there was it was found the envelope was immunogenic, and there was a lot of testing, but it didn't make it past phase 1 trials for quite a while. In 1998, VaxGen launched the first large-scale phase 3 trial of a recombinant GP120, despite participants in phase 2 of the trial actually becoming infected. In 2003, the RV144 famous trial was initiated, AIDVax and canarypox, a prime boost approach, and this is still the only vaccine trial that has shown any evidence of efficacy. In 2004, the STEP trial using a recombinant adenovirus vector, where there was a switch from wanting to generate antibodies to wanting to generate cellular immunity against a virus. So that trial made it through phase 2. 2009, it was the results of the RV144 showed modest success. It wasn't due to neutralizing antibodies. Some people think it might have been antibody-dependent cellular cytotoxicity, but they don't really know why that vaccine was successful. Since 2010, there's been a lot of focus on broadly neutralizing antibodies, and they've become primary HIV vaccine targets, but it's so far been impossible to generate broadly neutralizing antibodies with a vaccination approach. So there's ongoing work with mosaic vaccines, VSV vectors, virus-like particles that one of our speakers is going to discuss, and a new mRNA vaccine. I have underlined the 2001 nonoxynol microbicide trial and the STEP trial because even though these trials were unsuccessful, we may have learned some important things from these trials. The nonoxynol microbicide caused inflammation at the site of exposure to virus, and there was a signal that there was actually an increased rate of infection in the group that received the microbicide. In the STEP trial, there was an increase in infection in a group of people who received the vaccine compared to people who received the placebo. And that's what was interpreted as activated cells at the site of exposure to the virus, making people more susceptible to infection. So that's generated a new approach, a novel approach to prevention, where the environment in which you're exposed to the virus is controlled to prevent and reduce inflammation. We're also going to talk about the hepatitis C virus, and that was discovered in 1988. It's been also a huge problem to develop a vaccine that works against hepatitis C virus. There's only been one large-scale trial that was unsuccessful. There's no really good animal model for it, so it's a difficult task, as well as even without latency and the integration. Our first speaker, Dr. John Law, is going to tell us about progress towards a hepatitis C vaccine. John did his PhD at the University of Alberta, He then did postdoctoral work at Rockefeller Institute with Charlie Rice, and he's worked since then at University of Alberta with Michael Houghton, the discoverer of the hepatitis C virus, and recently he's moved to Memorial University of Newfoundland, where he's an independent investigator, assistant professor. So welcome John to his first CAHR meeting, and he's going to tell us about the hepatitis C, progress towards a hepatitis C vaccine.
Thank you, Mike. Thank you for the organizer, and thank you for all of you who show up early in the, to these sessions. I'm not a morning person. After this is a great opportunity to kind of share my work, and this is my CAHR, as Mike mentioned. After yesterday's with Dr. Husband's talk, I think it's very inspiring. So I'm just going to talk about a little bit about the Hep C vaccine work. So it will be slightly different than what you guys have been used to. This is my disclosure. I tend to talk long, so I try to acknowledge people first, and so that I don't kind of run out of time. And so on my left, I have a small group of people that's starting in the lab, and some of the students are looking at the longevity of the vaccine-induced protections. But most of my work that I'm going to talk about, it's work that I work with Mike in University of Alberta, giving you a little update about sort of where we think that the vaccines work we've been working. We also have collaborations with Stanford University, led by Stephen Fong, that we try to develop a vaccine that's induced both T cells and B cell response. Actually, seeing Mike's history of the talk previously, I think it's HCV vaccine development is mirror with the progress of HIV, except there's not as many clinical trials happen yet. I don't think I need introductions about hepatitis C virus, but I'd like to point out a few things that we can sort of relevance to this discussions. HCV infections cause chronic liver disease and eventually leading to irreversible damage, require liver transplantations. With the advance of the DAA, now the rate of liver transplantation is slowly decline. Interestingly, for Hep C is that there's about 20 to 30% of people that infected with hepatitis C virus are able to spontaneously clear the virus. I think for the last 30 years since the discovery of the virus, I think a lot of the people have been working on and try to define the immune correlate that allow the clearance of the virus. And I hope that the vaccine can induce similar types of vaccine immune response and therefore remove the virus from infections. And similar to HIV, people will ask you what is the likelihood of having a vaccine for hepatitis C virus. And in fact, actually, many people that are exposed to hepatitis C virus are susceptible for being reinfected, suggesting that that gives people some possibility thinking that it's difficult to have a HCV vaccine. But we actually see, if you're looking into a detail, this is one of the trials looking at people that are exposed to hepatitis C multiple times. What they notice is that in the subsequent reinfections of the virus, the clearance rate of virus jumping from 30% to 80%. And also, if you're looking at the length of the viremia and then the level of viremia, suggesting that in the secondary infections, the immune system actually learned something and able to have slightly better control of the virus. And so that provides some encouragement that vaccine is actually possible. And this is, I think, one of the concepts that I really want to bring it up in terms of the Hep C vaccine field is that many times when you're thinking about a vaccine, you're thinking about sterilizing immunity, where a vaccine provides protective immunity to prevent actual virus infection. But for Hep C, maybe this is not high bar that we require for the vaccine. And because Hep C is a chronic infections, most of the damage is at the chronic phase of infections. And perhaps for a successful vaccine, what we need is a protective immunity, where it's where actually the vaccine can just reduce the level of chronicity, it will be a successful vaccine for hepatitis C virus. Sort of similar to HIV view, the protective immunity of many clinical studies has suggested that we need both cellular and humoral immunity to provide protections. But for the purpose of the talk, I'm going to just focus on our efforts to induce neutralizing antibody for protection. Just kind of give you a few slides just to tell you why neutralizing is important for HCV control. In a single cohort study where they've followed people that's exposed to HCV, they look at difference between people that able to resolve hepatitis C on the left and people that eventually turn into a chronic infection. What they see is that for people that able to resolve hepatitis C virus, they notice that early inductions of neutralizing antibody is important. And while there's evidence of people able to develop neutralizing antibody at the chronic phase of infection, we think that the virus at that time already get a foothold, and therefore neutralizing antibody is not as critical. So early inductions of neutralizing antibody is important for protection. And also in a clinical study looking at people that's able to resolve the infections at the bottom, and also people that persist infections in the top. When they're looking at their level of breadth of their neutralizations, if you're looking at each of the row is at different patients, and each of the column is actually different variety of HCV PP that we established in the field as a panel to look at cross neutralizations. And if you focus on the graph on the right, people that are able to clear HCV infections, their antibody seems to be able to neutralize a higher breadth of different variety of HCV-CC found around the world, suggesting that besides in the timing of the inductions, the quality of these neutralizing antibody is actually correlate with protection as well. This is one of the slides that maybe it really kind of highlight the problem of HCV vaccine development. This is a slide provided by Stuart Ray from Johns Hopkins, basically looking at the diversity of different viruses. On the left, as you can see, is HCV, and then the diversity is even higher than HIV. And I like to show this slide to students in the lab because we know a lot about the diversity of the SARS-CoV-2 and the bottom right corner. If you actually put them in the same scale, it's actually just a dot on this slide. So HCV is actually a highly diverse virus, and the problem of having neutralizing antibodies is relatively difficult. This slide, it's just to show you that the regular entry pathway for hepatitis C virus, it's a multi-step process. It requires 4 different host receptors to allow the virus to enter. While the virus is highly diverse, they still require to interact with the same 4 receptors in order to gain entry of into the cells, suggesting that there may be some conserved area that we can target to have an antibody that can neutralize many different clades of the virus. The field is catching up to understand the structures of the glycoprotein on the surface. This is atomic structures of E1, E2. If you can see that on the left is the blue is the E1 proteins. On the right is the E2 proteins that divided into different color, showing different antigenic regions of the virus. Most of the antibodies response are targeted to the E2 proteins, and then the purple on the right-hand side is the neutralizing phase. This is the area that actually interact with one of the major host receptors, CD81. The reason I show this slide is basically show you that there is evidence of example of broadly neutralizing antibody. I think the key is to how to induce these antibodies by vaccine. Classically, most of the people using virus isolated to induce neutralizing antibody, but Hep C is a polymorphic virus. Actually, this is purified from Charlie's labs that the virus have slightly different sizes, and also the the assembly pathway is highly associated with the lipid machinery. So if you're actually looking at EM pictures, a lot of these viruses are decorated with apolipoprotein. So this is probably one of the ways that HCV is used to hide from the immune system because the E2 is not highly exposed on the surface. So in order to circumvent this problems, we actually, instead of using the glycoprotein, just the GPE1 and GPE2 to express in CHO cells, using the recombinant proteins to induce neutralizing antibody. This is just to confirm that the conformations of these proteins are recognized by conformation-specific antibody. We have tried to purify E1, E2, the heterodimers on the surface, or just the E2, which is the immunodominant targets of HCV virus. And on the top, these are E2-specific antibody, and on the bottom is either E1 or E1, E2 specific antibody. As you can see that both proteins are able to recognize by E2 proteins, and only E1, E2 are recognized by E1 or the E1, E2 antibody. And then subsequently, we actually test their breadth of their immunogenicity in the guinea pig model. On each of the column is represent a panel of different HCV PP that we actually isolate from different, that's established in the field, that are able to test broad neutralizations activity. And as you can see that each of the colors showing the warmer the color, reflective of higher neutralizing capacity. What you can see that in the E1, E2, they're able to show a higher broad neutralizing activity compared to E2, suggesting that presence of these heterodimers is important for the immunogenicity of the protein. In fact, at the time when the chimpanzee, the only animal, immune-competent animal model was present, this E1, E2 are able to support, show some efficacy to reduce chronicity. I want to highlight you at the bottom of the right-hand corner, where the animals immunized with E1, E2, while the vaccine is not able to reduce significantly the acute infections, but the vaccinated groups only have 16% progress into chronicity compared to the non-immunized group, which have 63% of chronicity, suggesting that immunized with E1, E2 is actually supportive to confer protection to reduce chronic rates in Hep C infection. This is actually subsequently tested in a human phase 1 clinical trial. The E1, E2 is actually immunogenic in humans and able to induce seroconversions, and you see evidence of T cell response in the trial as well. When we're actually looking at these anti-sera, we're able to see, demonstrate a certain level of cross-neutralizations against many different genotypes of the virus. The rainbow color represents different genotypes that you see in the earlier graph with different clade of HCV. From a single genotype of antigens, we're able to see evidence of cross-neutralizations against many different genotypes of the virus. Some of the genotypes are harder to neutralize, but we see evidence of that. That's give encouragements of potentially working on the vaccine. But when we're actually looking at the effect of antigens, on the left, we derive antigens from 1A virus. We're making E1, E2, or E2. If you focus on the red bar, antigen derived from 1A virus are able to confer protections against the 1A virus on the left. But antigens derived from 2A antigens do not provide these protections to 1A virus. Conversely, antigens derived from the 2A antigens are able to confer protections against the 2A virus on the right, suggesting that there might be some sort of genotype-specific determinants affecting their protections against the virus. So as I told you, there's many different genotypes of the virus spread throughout. I show you that our genotype 1 vaccine able to neutralize 1, 4, 5, and 6 really well. And then in the second slide, I show you that genotype 2 protein seems to only recognize genotype 2. And perhaps one of the things that we could think of is that we can multivalence the vaccines to kind of cover many different genotypes to able to overcome the diversity of the virus. But more recently, there's many more work in showing that the genetic differences between viruses might not be quite the determinants for their neutralization profile. More work still needs to be done. One of the take-home message I want you to take today is that vaccine for Hep C is possible. We are trying to using recombinant glycoproteins to induce neutralizing antibody, but we still think that there might be other effects that is important to induce cross-neutralizations. One of the encouraging fact is that recently there's many more platform of HCV vaccine is available. I think you will hear many more efforts of this front in the future. And thank you for your attention.
Thanks, John. So our next speaker is Dr. Eric Artz. Eric did his PhD with the legendary Mark Wainberg at McGill, and then he went to Case Western as a postdoc and stayed at Case Western for a few years. Came back to Canada in 2014 as a Tier 1 Canada Research Chair in immunovirology, and he's a full professor at Western since then, doing some great work, and he's going to talk about unconventional approaches for development of an HIV preventative vaccine. Eric.
Thanks, and good morning. Thanks for making it up. It's always good when there's more in the audience than there's up here, so I'm happy about that. All right, now, how do I move? I move this way. Okay, so I'm going to actually present a study that attempts to deconvolute what we observed in a macaque-based study where we looked at various vaccine candidates and actually saw some fairly effective responses and protection against subsequent challenges. But one of the complications was that we put so much into this vaccine that we really are now struggling to understand what was providing the protection. So the way we started this vaccine approach was to kind of understand aspects related to transmission of the virus. And from there, we were trying to develop a vaccine based on transmitted founder viruses to get those viruses that might be the ones most transmitted to provide us the immunity to prevent new transmissions. So as you probably all know, you start off with a large quasi-species, large population from a donor that gets transmitted to a recipient. And in that process, there's a huge genetic bottleneck, and as a consequence, only one strain typically gets through normal heterosexual or men who have sex with men exposures. And in that situation, we recently found that that process of the genetic bottleneck occurs almost mostly at the point of inoculation into the individual. So when it's in the female genital tract, you have a genetic bottleneck that occurs from the vaginal cervical mucosa to the blood, leading to systemic infection. The other thing that we had been studying on transmitted founder viruses is there any advantage of those viruses in terms of transmission efficiency as compared to regular chronic viruses that an individual would be exposed to? And one of the things we discovered was that, yes, indeed, what we were observing was that the chronic viruses, when mixed with transmitted founder viruses and applied to human cervical tissue, those viruses tended to get trapped in the cervical tissue, the chronic viruses, and the ones that snuck through to establish infection through the lamina propria and then with migratory cells infecting T cells were the ones that were designated transmitted founder or acute viruses, more often than not. So the one thing that we were trying to determine is what is different. So as many of you know, there's really not a lot of difference between, in a genetic sense, between, in particular, the envelope glycoprotein of these viruses, whether they're coming from chronic infections or they establish acute transmitted founder infections. And one of the things we did notice is that there was a differential binding to C-type lectins between these viruses, and that there was a tendency to have higher C-type lectin binding affinity when you were coming from a chronic infection. So we discovered all these things, and we said, well, let's try to determine if we put a transmitted founder virus into a vaccine construct, whether this provides any advantage. But before we did that, we wanted to try to understand what was it about the transmitted founder virus. If it's genetically similar to the chronic viruses, what is it that related to a higher transmission fitness? So we did all of these infections, we looked at different modes of transmission, and we looked at whether there was a difference between the mode of transmission and what type of virus got through. And what we observed is that people that got infected through injectable drug use had a tendency to have viruses that, not to a big surprise for many of you, that were very similar to what was in the population as chronic viruses. And the ones that were sexually transmitted had a tendency to have higher fitness in mucosal tissues to be transmitted than for systemic infection, similar to what I just presented. So we then went a little further again, because we know that there's not a lot of genetic differences we can attribute to this. And instead, what we did is we worked with Laura Mahal in University of Alberta to pass the viruses through a lectin array to try to see if there's any difference in glycan composition. So I'm not going to go through all this data. I'm only going to summarize. But suffice to say that if a virus is derived from a chronic infection, they have a tendency to have glycan content that is more in the high mannose range, that resembles what you would say would be similar to viruses or pathogens that are glycosylated with high mannose content. And that provides an escape mechanism to a lot of neutralizing antibody activity, for example. Whereas when you look at the transmitted founder virus, what you find is glycan content that's very similar to cellular proteins. So it's almost like mimic those cellular proteins to hijack a mechanism to enter into the cells and to establish an acute infection. And this was, again, looked at not only by lectin binding arrays to determine the glycan content, but we also did mass spectrometry analysis with Carol Krusney's lab, where he interrogated about 70 different glycans at different exiting points in the glycosylation pathway, whether it is in the endoplasmic reticulum, medial, trans-Golgi. And one of the things that's very notable is that when you look at host proteins, majority of them, about 80 to 90%, exit the glycosylation pathway at the very end in the trans-Golgi. But when you look at glycans that are found on viruses, what you see is that they exit glycosylation much earlier in the glycosylation pathway. So a lot of them have these, what I would say, not completely processed glycans that go through the glycosylation pathway. And this is even more so when you look at viruses that are present in an infected individual as compared to the ones that are transmitted that resemble more of the host glycans. I'm not going to go through this data, but suffice to say, there's also differences at the specific end-link glycosylation sites in envelope. So you see these huge genetic differences or glycan content differences and composition differences at specific end-link sites that we're only now trying to study to see how they affect transmission. And one of the challenges for us now is knowing kind of the glycan content associated with transmitted founder viruses at different end-link positions is trying to reproduce that for a vaccine. And you can imagine the challenges there, because it's not simply you add the construct and you hope to get that same glycan content, especially when you're producing in cells that aren't associated with transmission. So this has been a challenge, and Sashini, who's presenting, will kind of discuss how we're doing this with modifying the glycan machinery. So the other thing I want to address is another aspect that we put into our vaccine construct. So we were using transmitted founder viruses, but we also made this discovery years ago of this rare mutation in the CD4 binding site that appears with Maraviroc resistance. And Maraviroc is an entry inhibitor. And its mechanism of resistance was actually a very high binding affinity to CD4. It never actually appears in an infected individual. And what we think is happening, based on this observation and many others, is that this particular mutation induces hypersensitivity to broadly neutralizing antibodies that target the CD4 binding site. So one of the hypotheses we had was, well, if you elicit B cells with precursor or germline BCRs that may lead to a broadly neutralizing antibody, and you have the virus that induced it hypersusceptible to that broadly neutralizing antibody, it becomes a closed loop. So you will never induce that in a natural infection. So we said, well, in a vaccine, this might be profitable. I think I'll just skip this if I can. So we tested this particular mutation in mouse models, and the trouble with mouse models in our hands were simply that the human CD4 and the mouse CD4 doesn't resemble itself enough to induce any binding affinity that might induce a conformational change that locks it in a position that it can elicit these broadly neutralizing antibodies. So in this case, we use CD4 mimetics to induce this conformational change and vaccinated with a virus-like particle to see if we could get this. And lo and behold, through prime boost immunizations, we could induce these precursor B cells that are targeting CD4 through their BCRs. And we can even select now and do single-cell sequencing, which we're doing at the moment, using this particular mutations. And we get an enrichment of those B cells that have BCR specific to the CD4 binding site. Now, whether they go on to produce neutralizing antibodies, I can't say yet, but this leads us to the last couple slides where, this all is done in retrospect, because what we did is we went forward with a vaccine trial in macaques and tried to attempt to test everything together. And so what we had done with a large team, Keith, led by Gary Kobinger, so Keith Folk, and then a number of people that were forming this team, we developed all these constructs on a VSV backbone, where we used the A74, which is this transmitted founder virus. We made all of them contain the N425K mutation, and we tried to control for glycan content as much as possible. In order to enhance expression of the envelope on the VSV particle, we played around with the transmembrane tail and the intracytoplasmic tail of envelope, and we came up with a solution of using the SIV envelope tail to enhance expression of the HIV envelope. And what I'll show you is we did a prime boost immunizations with a whole lot of different combinations, which also confused the situation quite considerably. But what we observed through these multi-challenge studies, so we did 7 low-dose challenges, that in one group we had about 60% protection. Now, that may not seem that dramatic, but this is a protection across subtype challenge. So this level of protection has actually never been observed before in a cross subtype challenge. So we immunize with a subtype A envelope, and we challenge with a subtype B virus. And one of the things we noted in this study, not dissimilar to what the Ad5 vaccine had showed us, is that you get a big induction of CD4 positive T cells with the VSV vector. And if you use the VSV vector in that configuration, especially on a CTL basis, that level of CD4 activation will lead to infection, and that's what we observed in most of the other groups. But when we did an immunization at the end where we didn't induce the same level of CTL activity at the last immunization, that's where we saw the protection. And one of the problems is we challenge way too soon after the immunizations. So the other thing we observed was that 60% of the protected group actually had neutralizing antibody responses to the challenge virus. So that's something also that had not been observed. So sorry for the very rapid sort of discussion of all the things that went into this. One of the things we're doing now is taking a step back. We can't basically test every aspect of the what I would call semi-successful vaccine and try to determine what was the most potent aspect of it. So instead, we're working with Lisa Wager at UC Irvine, who's developed this nice tonsillar organoid model for vaccinations, which has been quite successful. And you can actually induce antibody responses, even get BCR selection, et cetera. So we're doing that right now to test a whole cadre of vaccines with all these elements introduced to try to find the best immunogen that would go forward into the next macaque studies. So I'll leave it there, and thanks a lot.
Thanks, Eric.
Okay, so now we're shifting a little bit more into the prevention side that Michael mentioned. So our next speaker is Lyle McKinnon. He's an associate professor in the Department of Medical Microbiology and Infectious Diseases at the University of Manitoba. His lab focuses on mucosal immunology and STBBI risk in key populations in Southern and Eastern Africa and Southeast Asia. He's published over 130 peer-reviewed publications, and today he's going to share with us some new work that he's working
on. Thanks, Amy, and good morning, everyone. So my lab is really interested in mucosal immunology, and I really like this concept of a firewall. So, every mucosal tissue has to interact with its outside environment, and you have multiple layers of immunoregulation that that make sure that you don't get excessive inflammation in most cases. There's a lot of interest in inflammation in health research, and I think this perspective in science was really interesting. That inflammation is part of a natural physiological process, and it's only where you have excessive inflammation that this tends to lead to problems. So the same machinery that leads to inflammation is also going on in the background in homeostasis. Of course, many people in the audience will know that HIV benefits from inflammation at all phases, so during pathogenesis, during treatment, and I'm going to talk about during transmission. So when I was at Caprisa in South Africa, we showed that a graduated cytokine score was associated with HIV risk. So the more elevated cytokines people had, the more increased risk there was. And this table basically shows, if you have 3 or more cytokines elevated in genital secretions, you have increased HIV risk. And as the number of cytokines elevated increases, you have more increased risk. So this is dose dependent. So basically, there's sort of a gradient of increased risk that's associated with having more inflammatory cytokines elevated. And the cytokines that are in this score are sort of the inflammasome type cytokines like IL-1 beta, TNF alpha, and others. And recent data from the VOICE trial has sort of validated this in an additional cohort. And just to also make the note that this is sort of subclinical inflammation. So some of these women will have overt clinical signs of inflammation, but many of the women in this study would have no symptoms at all. So it's a subclinical definition of inflammation. So how we think this is working is, this is a sort of a cartoon of HIV transmission. So the virus has to get across the mucosal barrier, has to find target cells to infect, and then has to disseminate into the systemic compartment. And inflammation probably just increases the odds that this happens or increases the rate at which it happens. And so we've shown that pro-inflammatory cytokines are associated with HIV risk, and these in turn are associated with biomarkers of the mucosal barrier. So when you have a breakdown in the mucosal barrier, you have more inflammation and vice versa. And this also probably leads to an increase in HIV target cells and immune cells of all kinds. And we have this is old paper now, but basically showing that all of these things are highly associated with each other. And we have a follow-up paper that shows that this is true if you look at longitudinal samples. So in the same analysis of this Caprisa cohort, this is sort of the beginning of the story I'm going to talk about today, we also found that if you look at individual cytokines, some cytokines were associated with decreased HIV risk, and one of the cytokines of interest was IL-2. So this made us think about IL-2 and regulatory T cells, and that's what I'll be talking about today. So IL-2, Tregs constitutively express CD25, which is part of the trimeric IL-2 receptor. And one of the things that Tregs do is actually consume IL-2 out of the environment. So IL-2 can also stimulate CTL responses and all kinds of inflammatory type responses. And IL-2 regulates this in part by consuming IL-2 from the environment. So there's a lot of work going on right now in terms of IL-2 engineering. So in cancer field, people don't want Tregs, so they try to make IL-2 preferentially bind the dimeric receptor, which leads to the sort of CTL type response and all the other associated responses. And in autoimmunity, people are really interested in engineering IL-2 so that it induces the regulatory response, so suppresses inflammation. So we were really interested in this question. So does IL-2 induce genital Tregs? So we're interested in the genital tract because we're looking at HIV transmission in women. And so does IL-2 induce genital Tregs that can regulate inflammation during inflammatory exposure? And we did this. So on the far right of the slide is my postdoc Al, who led a lot of the human work. And then Thomas Maruka and Marina Costa Fujishimi really helped out a lot with the mouse work. And then Faisal is my PhD student shown in the middle there, and he led the mouse work in my lab. So there's a lot of interest now in tissue Tregs, and there's a lot of evidence that these are different than the sort of Tregs you study in the immunology textbook. And this was a really neat paper that came out from Andrew Liston's lab, showing that if you look at the gene expression in tissue Tregs, it's quite different from systemic Tregs. And actually, these Tregs are kind of temporary resident Tregs. So they circulate, every few weeks. They establish transient residency, but they're very distinct. So most tissues are distinct from Tregs are distinct from gut Tregs, which are in turn are distinct from lymph node Tregs and Tregs in systemic circulation. I won't go into detail, but a lot of the literature around Tregs is focused on reproduction. So Tregs are really involved in reproduction, in sort of avoiding rejection of the fetus, but not much. Fewer data are available on Tregs and infection. So we did a cross-sectional study in the sex worker outreach program where we work a lot in Nairobi. And this is a really large PEPFAR program, which I think is still funded, hopefully. This program delivers HIV treatment and prevention to a large population of sex workers and other key populations at risk of HIV. And we use this to embed research studies. So we developed a flow cytometry panel on fresh cells in Nairobi from cytobrushes. So cytobrushes collect cells from the endocervix, which is shown on the left cartoon. So that's the entry to the uterus. And using this sample type, we could define Tregs as being C25 positive, C127 negative, and we showed that most of these cells express FOXP3. And we looked at a few other markers like CTLA4 and other Treg-associated markers. We found that Tregs were more frequent in the cervix than the blood, but there was a correlation between compartments, and we've seen this with lots of different immune cell populations where there are certainly differences between the cervix and the blood, but you find usually a weak to modest correlation between the frequency of cells in the blood and the frequency of cells in the cervix. So what we showed in this paper that came out a few years ago now is that women with more genital tract Tregs had decreased inflammation. So this is true if you looked at a number of different cytokine and chemokine concentrations. The the people on the right side of these figures have the most Tregs, and you can see that they have the lowest concentrations of things like IL-8, interleukin, IL-1 beta, so all of the same inflammatory cytokines that we showed were associated with HIV risk. Also of interest is that women with more Tregs had a lower abundance of CD4 cells, Th17 cells, and again, so the Treg tertile 3 was associated with the lowest concentration of these HIV target cells. And if you're this was a cross-sectional study. If you're interested in more details, I do have a poster apparently somewhere where I'll talk a bit more about trying to confirm these data in a longitudinal cohort. So I'll just finish by talking about a mouse study that we did. So we wanted to model this in mice, and so we tried a number of different ways of delivering IL-2, but we ended up doing IP dosing of an IL-2 conjugated to a monoclonal antibody. And people have shown previously that if you conjugate IL-2 to this monoclonal antibody, you preferentially induce Tregs. And there's another monoclonal antibody that you can conjugate IL-2 to that preferentially increases NK cells and CTL responses. So basically, you dose the mice with progesterone, you dose them with whatever IL-2 construct you want, and then you measure Tregs and then can do other experiments downstream. So one thing we showed here is that if you do the IP dosing of IL-2 with this antibody that induces Tregs, you indeed induce Tregs in the genital tract. So if you look at the figure in the middle, the PBS mice have a few IL-2. So the Tregs in these mice glow green because of GFP linked to FOXP3. And so you can see that the IL-2 dose mice, both by flow cytometry and by imaging, have increased frequencies of Tregs in the female genital tract. And then the Tregs that were induced also expressed increased levels of some of these Treg-associated markers, like CTL3 and GITR and ICOS. In some cases, these are significant. In some cases, they're not. We also show that I mentioned before that there's different IL-2 antibody conjugates. So basically, if you want more NK cells or more CTLs, you can use the IL-2 conjugate to the antibody that induces those cells. But what we showed is that the IL-2 conjugated to the antibody that induces Tregs preferentially induces Tregs without inducing many of these effector cells. So you can sort of fine-tune what kind of IL-2 effect you get by choosing which antibody. And basically, this changes the way the cytokine interacts with the receptor. So the antibody that we're using makes IL-2 interact with the trimeric receptor, which is the one that's preferentially expressed on Tregs. So what we wanted to do is ask the question, if we induce Tregs, do the mice with the increased Tregs have less inflammatory response if you induce an exposure that leads to inflammation? So we used two different stimulants here. So nonoxynol-9, which Mike talked about in the intro, which is an inflammatory microbicide. And we also used a sort of BV-associated bacteria called Mobiluncus muliaris. So in the nonoxynol-9 challenge mice, so basically in this experiment, what we do is we dose the mice, increase the Tregs, and then give them the inflammatory challenge. And what we found in the nonoxynol-9 challenge mice is that they had increases in neutrophils. But so there's two different readouts in this experiment. One is genital tract neutrophils, and the other one is leakiness of the mucosal barrier. So nonoxynol-9 has been shown to increase leakiness of the mucosal barrier, and inducing Tregs didn't really stop this from happening. So, but it did prevent neutrophils from coming in. So if you just give nonoxynol-9 without Tregs, you get a big influx of neutrophils. And the Tregs that are induced in the genital tract were able to prevent these neutrophils from coming in. What was interesting in the Mobiluncus challenge mice, so the same experiment where you induced the genital Tregs and then challenged with Mobiluncus, here we found that basically the Tregs were able to prevent the neutrophil influx and also maintain the mucosal barrier. So, if you give just the inflammatory bacteria, you have a lot of leaking of the dye into the lamina propria, and Tregs were actually able to stop this leakiness of the dye, and also to stop neutrophils from coming in. So basically, they're doing the anti-inflammatory type things that you think that they should be doing. This is the last data slide. So we showed there's a type of mice called dereg mice, which you can treat with this diphtheria toxin, which knocks out the FOXP3. And we are able to basically show that these effects were FOXP3 mediated. So you need FOXP3 in the system to have the effect of this IL-2 antibody conjugate. So just in conclusion, there's a dose-dependent relationship between genital inflammation and HIV risk. And 3 cytokines, including IL-2, showed protective effects in a longitudinal clinical cohort in South Africa. Genital Tregs inversely associated with genital inflammation in women from Nairobi, and we're expanding this in Uganda. If you IP dose mice with IL-2 conjugated to a certain monoclonal antibody, you can selectively increase genital Tregs. And then mice with these genital Tregs are able to control inflammation and maintain barrier integrity in a FOXP3-dependent manner, and probably in a mechanism that we think probably involves neutrophils. So just in a big picture, there's lots of different causes of genital inflammation, and genital inflammation has a big effect on women's health. So not only infectious disease outcomes, but also reproductive health outcomes. So this is a slide borrowed from some colleagues at Caprisa. So there's lots of different things that stimulate inflammation, which would be considered the match. And then there's lots of different strategies you can use to try to control inflammation, and it's really hard to control all these different sources of inflammation. So targeting sort of immunoregulatory pathways is a way of sort of trying to counter all these different causes of inflammation all at the same time. We have a lot of future directions. We're trying to understand mechanisms. We're looking at IL-10. We want to look at humanized mice. We want to look at other infections like HSV2. And also, can we make this more of a tissue-targeted delivery? And there's been some really interesting work in sort of the bioengineering field, and I've learned a lot in this project about cytokine engineering. So I think there are some potentials to do some local delivery. I think where does this fit into HIV prevention? I think controlling genital inflammation is broadly relevant for women's health. I don't think it's going to replace PrEP really, but it could be something that augments some of these things. And I'll just stop there because I'm over time, and thank the people involved in the study. Thank you.
Okay, our last speaker today is Dr. Chara Kaushik. She is the scientific director of the CAHR Institute of Infection and Immunity and a professor in the Department of Medicine at McMaster University. Dr. Kaushik has spent 25 years running an interdisciplinary research program in women's reproductive health, specifically basic, clinical, and translational research, examining susceptibility and immune responses to sexually transmitted viruses such as HIV-1 and HSV-2. I think today we'll be seeing some of her recent translational work.
Thanks, Amy, and good morning, everybody. Sorry there's no coffee. I'm missing it too, so bear with me if we are all a little bit slower, but great talk. So thanks, everybody, and thank you especially to Lyle for setting up the inflammation in the vaginal tract or in the reproductive tract and its repercussions on susceptibility to HIV, because I'm going to talk a little bit about that, but I was hoping somebody else would do some microbiome introduction, but that didn't happen. So here we go. So this is an older slide, but I like to project it primarily. There's a lot of information on this slide, but I really want you to take away from it is that HIV is primarily a mucosal infection transmitted sexually. And I say that because on the left side of this column, there are all the different sites of transmission of HIV, and on the right side is the numbers in millions. And if you look at this, 90% of HIV is transmitted through genital and rectal mucosa, and 40% of HIV is transmitted just through female genital tract alone. So a big part of prevention could be if we can prevent transmission through the genital tract, we could prevent a huge number of HIV infections. And we know that globally, women are disproportionately affected by HIV, but also by other STBBIs. So any kind of protection methodology that we can use in the genital tract of women would lead to a significant benefit for not only HIV, but for other multiple STBBIs. So a quick 101 on vaginal microbiome. I'm sure most people in this audience would be familiar, would have heard about it. So just the high-level recaps. So vaginal epithelium is lined with bacteria. There are trillions and trillions of it. Vaginal microbiota that is dominated by lactobacillus species and has a low pH is correlated with less risk of STIs and beneficial outcomes of pregnancy. Whereas vaginal microbiota that is dysbiotic is associated with increased STIs and poor outcomes of pregnancy. Dysbiosis can lead to bacterial vaginosis, or BV, which is known to increase HIV infection risk by 2 to 5-fold. So we know that there are ethnic differences in vaginal microbiota. There's disproportionate effect in women who come from African and Hispanic backgrounds. So both the top and the bottom parts of this panel really just show that if you look at on the top panel, the yellow and the blue, which correlates with the lactobacillus species, compared to the African American women in one of the studies with the European ancestry just below it in the yellow and blue again, you can see the huge differences in the percentages of women that have this type of optimal microbiota in European ancestry. The bottom pie chart shows something very similar just in percentages. And you can see on the right two side, or the right two pie charts, that women who come from the Black and Hispanic have about 40% microbiota that is diverse and correlates frequently with dysbiotic microbiota. So higher issues with inflammation that correlates with dysbiotic microbiota. And this is just an introduction to show that the more diverse the microbiota, so on the right side, you will see the more diversity, the higher the likelihood of different kinds of STIs. So again, dysbiotic microbiota correlates with higher inflammation and higher rates of different kinds of sexually transmitted infections. Just a big quick 101 on hormones. So many of you may know that my lab and many other labs have focused a lot on looking at the effect of hormones, because everything that happens in the reproductive tract happens in the context of hormonal regulation of the physiology, but also of the immune responses in the female tract. And without getting into decades of information, just big takeaway messages are that in general, estrogen is protective against HIV and SIV. This has been shown in vitro, in macaque models, in clinical studies, whereas progesterone in general increases susceptibility to HIV and SIV. And I won't get into progestin-based hormonal contraceptives, where we've done quite a bit of work, and there's a lot of clinical data that correlates with increased susceptibility to HIV. So just a quick high level, we've done lots and lots of work to show estrogen is beneficial in many different ways. It increases antiviral responses, innate responses, CD4 T cell responses. And we've done this through looking at in vitro studies, in vivo studies, and clinical studies that we're starting to do now. So what I'm reporting today is just the outcomes of a single phase 1 clinical trial, which based on the decades of work that my lab and others have shown is that estrogen is beneficial and could lead to protection against STIs. The thinking was that could we design a methodology for prevention where we could combine local estrogen, because we know estrogen in systemic is not something that is preferred due to cardiovascular risks, but could we do local estrogen and combine it with probiotic treatment to see if we can improve vaginal health? So the study design is shown here on the left, and I'm not sure how well you can see it, but basically it was a 30-day intervention, 30-day daily doses of E-string, which is a low-dose estrogen ring that women place in their vaginal tract. It's used for menopausal women, but this was off-label use, so it was Health Canada approved trial. And combine it with Represh Pro B, which is a probiotic that combines rhamnosus, lactobacillus rhamnosus, and lactobacillus reuteri. So women were given this for 30 days, and they had to come and give samples at baseline, mid-intervention, at the end of intervention. A smaller group of women agreed to give a sample 1 week after. We wanted to see how long the effect of probiotics in this combination would last. So when I show you data, mostly we're comparing visit 2 and 4 as the beginning and end of intervention. And we looked at a number of things. I will not be reporting today the safety, feasibility, and tolerability data, which has already been published, and we exceeded all the targets that had been set for this in those parameters. What I'm showing you today is the analysis on the biological end of things. Did we really enhance lactobacillus abundance in the vaginal microbiota? Did we reduce cervical vaginal inflammation? So those are the two things that I will be showing you today. So as I said, the safety, feasibility, and tolerability has already been published, and I want to acknowledge the students and postdocs who contributed to it. Today I'm talking mainly of the biological outcomes. So do these interventions improve vaginal health? And I want to start out by acknowledging Smriti Dabi, who is a postdoc who is actually currently situated in UBC and has been collaborating on the analysis of the data since the trial was finished in 2022. And a new postdoc in the lab, Mesa Niazi, who is a bioinformatics specialist and has done a lot of this analysis. So without getting into too many details, this is just a demographics of this. There were 4 different interventions, so you will see a lot of the data is divided into 2. Because the trial was done during COVID, we had to stop and start multiple times. So we just didn't have enough samples to do analysis by specific interventions. So the 4 interventions are eastring alone. Does estrogen alone do something? Combination of eastring with oral probiotics, combination of eastring with vaginal probiotics, and then just vaginal probiotic alone. And if you look at demographics of these 4 different groups, there's really no difference in any of these. We looked at the data to make sure it reconciled with most of the other reported in literature, and indeed we see that the microbial community state types, when you sort out all the samples post sequencing, fall into the typical community state type 1, 3, 4, and 5, which is the smallest amount, and that is aligned with a lot of the data that's out there. We also looked at the alpha diversity, and as expected, CST type 4, which has the maximum diversity of microbiota, shows significant increases in the samples in terms of the diversity indexes. And the 3 different types of microbiota also sorted themselves out in a Bray-Curtis distance graph as well. One of the things that we noted, and you will see this, that is that we looked at to understand whether there is any seeding effect, because I was asked multiple times, like, is the vaginal probiotics leading to seeding? Is the oral probiotics leading to seeding? So the 2 different, this is the abundance chart for the 2 different probiotic species, and the answer is yes. Only in the vaginal probiotic administration, there is actual seeding of the probiotics that's there, and you can see that in visit 2 and 4, you can actually see those. There's a clear abundance of this. However, in the 5th visit, most of it had disappeared. So we looked to define, because we didn't have enough statistical significance or strength in the numbers in terms of looking at specific interventions, to just look at overall all 4. Was there an improvement in the lactobacillus? And we defined this by 4 different criteria: a transition to lower community state type, a shift from low lactobacillus to high lactobacillus in quartiles, a decrease in overall alpha diversity, and a decrease in BV-associated bacterial types. And the pie chart shows that about 51% of the women actually showed one of these or multiple of these 4 different positive changes, whereas 46% had no change. 1 person actually shifted from CST3 to CST1, which is actually a negative shift, but by the last visit or the follow-up visit had shifted back to CST3. If you look at all of these combined, you can see that in the 4 different categories, multiple overlaps occur. That there is CST shift, which is the hardest criteria to meet, is the lowest numbers, but majority of the women have decreased microbial diversity, improved lactobacillus, and species-specific shift. And then if you look at this, the microbial diversity on the women who had 21 out of the 41 women who had the shift, there's actually an overall change in the alpha diversity index in these women at the end of intervention, which is the 3rd bar chart that you're looking at. What was very encouraging was that in all of these women, the percent of lactobacillus, whether it was probiotics or any other kind of natural probiotics or lactobacillus, shifted to almost 87% by the end of intervention, and actually stayed pretty high even in the 5th visit to about 63%. When we looked at these and we categorized these into optimal and non-optimal based on the type of microbiota that was there. So when this was done, you can see this is from the 4th visit. We divided them into optimal and non-optimal, and the lactobacillus species are on the bottom. So on the right side of the optimal, you can see that there was heavy prevalence of lactobacillus as expected, and in the non-optimal, the BV-associated bacteria are heavily prevalent. And when we looked at, we had 43 cytokines that we measured in these cervicovaginal samples. And when we looked at these cervicovaginal samples in the same optimal and non-optimal categories, on the left side, you can see, as Lyle had shown, the inflammatory cytokines were all up. And on the right side, in the optimal categories, these are some of the non-inflammatory cytokines or anti-inflammatory cytokines that were up. So we analyzed this data based on inflammation scores, which Lyle has also talked about, so thanks, Lyle. And when we looked at the inflammation scores of those who had positive change versus those that didn't, you can see a clear decrease in the inflammatory scores in the women who showed improvement. And when we do a cytokine ratio of inflammatory to beneficial cytokine ratios and compare that to the KO index, which is alpha diversity, you can see that there's a clear distribution of higher diversity correlating with higher indexes, so more inflammation. And then finally, the last piece of data that I want to show you is that did it have benefit to the clinical BV diagnosis? And the answer is yes. The same number, about 50 to 55% of the women actually showed benefit in the inflammatory as well as clinical BV diagnosis as seen by Nugent scores, and also in the CST4-based criteria for BV. So in summary, approximately 51% of the women showed an improved vaginal microbiota. Vaginal probiotics lead to seeding of lactobacillus. There's a significant decrease in diversity and increase in lactobacillus in women who showed positive responses. There's lower inflammatory cytokines in women with optimal microbiota. There's a significant decrease in inflammation scores from baseline to follow-up in women who showed positive response. And positive responses, changes in microbiota correlated with decreased inflammatory scores. And so our vaginal probiotics are better or with or without estrogen. So the big picture conclusion aligned with the theme of today's discussions is that probiotic treatment with or without estrogen can enhance optimal microbiota in the vaginal tract. Optimal microbiota decreases inflammation, and potentially it could then decrease susceptibility to HIV and other STBBIs. And the plan is to actually start planning a phase 2 clinical trials. Because of the decrease in numbers, we're going to really have to think about how to design this trial. Which intervention should we choose? Just the vaginal probiotics? Should we only go with women with BV because we see 50% of the women showed benefits? And whether we should have control group with no intervention. Again, my thanks to all the people who participated in this study, the Women's Health and Women's Hand Clinic, where the study was actually done, the CTN that helped analyze the data and supervise the study, and all the people who were involved in it, especially Smriti and Mesa, who did the analysis of the biological data. And with that, I'll stop.
Thank you, Charu. I apologize. The clock has struck 0, so rather than take questions, I'll just ask you to put your hands together once more for the 4 speakers who got through their excellent talks.